Thank you, Dr. Ellis. It's an honor to be here today and a real pleasure to be back in Houston. I started my career here at MD Anderson over 20 years ago, and Dr. Spitz was my chair and recruited me, and I see a number of friends and colleagues from a long time here, so it's a pleasure to be back. I stand between you and lunch, and of course we're talking about obesity. Seems like that happens a lot. I'm sure lunch is low calorie, so we have nothing to fear, but we will talk about this link between obesity and cancer, and particularly focus on lessons from animal studies, preclinical research. Margaret did a wonderful job of talking about the human side of this story, and what can we learn from animal studies is really the focus, and particularly what metabolic and molecular targets can we deconvolute from this association and begin to develop intervention strategies for breaking these obesity and cancer links. And the punchline will be growth factors and their signals and inflammatory signals, and really the interactions of these that are emerging as key targets for breaking this connection. And like Margaret, I probably got this idea from Margaret, but from my preclinical side, I've prevented a lot of mouse cancer in my career, and not that proud of that, actually. The key is translating these findings to the human. And as Margaret said, the process can be somewhat delayed by just lack of connection. In this, I think a transdisciplinary approach is critical to accelerate this process. And I'll tell you a little bit about how we're trying to do that. And that was one of the reasons that besides family, I'm a North Carolinian, so we really kind of needed to get back home, but also more opportunities at UNC to connect what we're doing in the laboratory with the clinic, and that was a strong appeal. So I'll talk about that at the end. We'll start with this cartoon from now over 10 years ago, and it says the shape of things to come. And as Margaret said, unfortunately, it's increasingly the shape of things today. We see this obesity epidemic. I was born in 1958 when the prevalence was around 12%. So just in my lifetime, the prevalence of obesity in adults has tripled. And we see now almost 7 out of 10 adults are in this overweight or obese category. Getting close to 40% are actually in the obese, and that's defined by a body mass index greater than 30. So weight over height squared greater than 30 is a kind of a crude measure for this, but it's a broad one that we use. And as I said, 4 out of 10 almost in this country. And an alarming, almost 7% are in this extreme obese category, BMI greater than 40. Now, we've known the impacts that this phenotype has on a number of disease processes. Certainly, it describes a state of metabolic dysregulation that we know it's links to insulin resistance, hyperinsulinemia. So prediabetes, diabetes certainly connected here. And we have the syndrome that we define as metabolic syndrome, and that's one of the hallmarks of that. Dyslipidemia is another. Increased waist circumference and hypertension. Those are the 4 kind of classic metabolic syndrome characteristics. But we also know that obesity, this obese phenotype, is associated with a pro-inflammatory state. So increased cytokines and chemokines. Not a huge acute type of inflammatory response, but a chronic smoldering response, but over many years can have a huge impact, and we'll talk about that. There are vascular perturbations, things like VEGF we've heard about are elevated in obese individuals, altered adipokines. A molecule I've been particularly interested in for a long time is insulin-like growth factor 1, which is a partner in crime with insulin, but really particularly dangerous as a strong mitogen, a strong proliferative and survival agent linked to cancer. And this is also elevated in the obese state, particularly the bioavailability of IGF-1 increases. So we've known about this connection with a number of chronic diseases, but really the cancer story has really emerged over just the last decade or so in terms of the breadth and depth of the problem. In fact, estimates, Margaret showed the doll and pedo, which said diet roughly was about a third, contribute to about a third of cancer, and much of that is obesity. And there have been some really nice analyses suggesting maybe about a quarter can be attributed specifically to this obese issue. And that's about 150,000 deaths per year in the United States. So a major problem. As Margaret said, it's, not every cancer is equally linked to obesity. This is from the American Institute for Cancer Research, which has a wonderful surveillance of the world's literature on this. If you're interested in this, you can go online to their website, and they have updated meta-analyses and systematic reviews that are really quite helpful in tracking the evidence along this line. But as Margaret mentioned, endometrial is the most common, was the first we recognized as an obesity-driven cancer. About half of endometrial cases have been attributed to obesity, but you see, on the order of a third to a quarter for many of these, and overall about 25%. We'll talk a little bit about pancreatic. I'll focus on breast just as a key example, but we'll also talk about pancreatic at the end, because that one's rising, and it's thought to be rising in prevalence as well as cause of death. It's predicted pancreatic cancer will be the #2 killer in the United States in the next 10 or so years, and largely driven is the thought. Lynn Matresian and colleagues have written elegantly on this, that obesity is really a key driving factor for this, and so something we'll wrap up with that one. I think a quite impactful paper was published just last mid-October from the Journal of Clinical Oncology, and it's the American Society of Clinical Oncology position statement on obesity and cancer. And it's interesting because I've worked on this for 25 years, and I've had colleagues after this paper was published come up and say, did you know that obesity and cancer are so strongly linked? This is a real big problem. I thought they knew what I was doing, but it took the oncologists to open their eyes, I think. But this has emerged as a key issue in the oncology field. And so this position statement, I'll just hit some of the highlights. They call it a central challenge in cancer prevention and care. It's the leading obesity is the leading preventable cause of cancer in the United States now. It passed smoking. These two together, as we've heard, account for a large proportion of the preventable cancers. And so we've got two targets here to really go after. Obesity not only increases the risk, that's really been the focus of most research, but it also increases recurrence, lowers survival. We have time to show data, but we see that it seems to impair the response to chemotherapy across a number of classes. In breast cancer, for example, endocrine therapy, cytotoxic agents. There seems to be an impairment of response, and that contributes to the poor prognosis that one sees in obese individuals. And prediction from this ASCO group was that by 2030, half a million Americans per year will be diagnosed with obesity-related cancers unless corrective action is taken. And so this is a huge, they call it a tsunami that's coming. And the impact on our healthcare system will be incredible, not to mention the quality of life of these folks if we don't do something about it. So that's the question, what do we do about this? And it's not just the United States, this is happening in the world. As we sit around sort of planning how to approach this problem, much of the emphasis thus far has been on preventing obesity in children particularly, and that, as Margaret mentioned, this is a problem. We're seeing, upwards to 20% of our kids overweight and obese already. That's a strong predictor of future obesity rates rising if we don't do something about it. So appropriate to focus on prevention in children, and I see several colleagues that are focused on that, nodding heads. But to me, the elephant in the room here as we talk about this is, what do we do about the folks that are already obese? And as we've heard, that's quite a few in the United States, and an estimated 720 million adults worldwide that are currently obese. What do we do about the cancer problem arising in this large population of obese individuals? And being a laboratory-based person who got to hang out with epidemiologists most of my career, I felt like a mechanistic approach was needed to identify targets and strategies to break these obesity and cancer links. And so that's what we went after. Margaret mentioned the hallmarks of cancer. This is a nice concept from Doug Hanahan and Bob Weinberg that captures the essential alterations in really all cancers. And And I don't have time to go into detail on this, but I can tell you that obesity impacts each of these hallmarks of cancer. And we'll focus particularly on altered growth signals and metabolic reprogramming that occurs with cancer. It's a switch, the metabolic aspect is cancers tend to switch their utilization of glucose away from making ATP towards making substrate for new cells, new daughter cells. And so that's a characteristic of cancer cells, and obesity feeds right into that, as well as the certainly altered growth signals, as you can imagine. Inflammation is the other biggie, and in my view, actually several of these other hallmarks, including sort of unresponse to death, what are typical death signals, but cancer cells can turn those off. And immune surveillance. We're going to hear about immune later this afternoon. Sustained angiogenesis, obesity feeds right into that as well. Limitless repetitive potential relating to really telomerase, reactivation of telomerase, and as well as enrichment of tumor-initiating cells. Obesity feeds into that. Tissue invasion and metastases, understudied. We're beginning at UNC to really focus on obesity metastatic links, but that seems to be there as well in genomic instability. Many of these, I think, spin off. There's an inflammatory component to many of these as well. So we'll focus on those top two. But I wouldn't say obesity, it's hard to say obesity causes cancer when you get right down to it, but it certainly is enhancing each of the hallmarks of cancer, making things worse almost across the board. And so, as we've heard, this is a strong proponent of cancer development, progression, spread that we really need to deal with. And so the other approach we've really taken is to use the mouse models and try to model. We've gotten quite good, I think, in the field of modeling human cancer in mice, particularly certain cancers. But can we bridge the energy balance question into these animal models where we've got altered key human-like alterations in key genes and pathways to pick this apart and sort out what targets do we go after, what can we do to break these connections. And I'll focus today on particularly the Wnt beta-catenin signaling pathway, important in a subtype of breast cancer, basal-like breast cancer. That's a particular challenge because we don't have targeted therapy. These cancers don't express the typical receptors that we can go after, such as estrogen receptor, progesterone receptor, HER2. So we don't have really much in our arsenal for these triple negative basal-like breast cancers, and this Wnt pathway really drives this basal-like breast cancer process. So I'll focus on that, but I will mention the RAS pathway with our pancreatic story at the end as well. And here's sort of a typical way we start our approach to this is we can make mice. And these are genetically engineered mice, typically, that we're using, that reflect a human genetic variant that's associated with cancer. And we can make these mice lean by restricting their energy intake, a term we call calorie restriction, but it's simply a low-calorie diet. And you can see they stay lean, they're less than 30% body fat. We can make mice obese as well, the diet-induced obese regimen, and an intermediate group here that's overweight. So we've got kind of a three, the spectrum of weights that we're trying to study and reflect from the human. And we were curious about particularly a panel of potential intermediates. And at this time, there was evidence arising that these insulin and insulin-like factors may be involved, so we looked at that. Leptin and adiponectin are both produced by adipocytes, have been shown to impact inflammatory processes as well as other survival pathways in cancer cells. So we looked at that, and of course IL-6 as sort of a candidate intermediary for the inflammatory response, as well as tumor volume in these genetically engineered mice. And what we find is that when the mice are lean and sort of maintain their kind of normal weight throughout, IGF and insulin are both low. Leptin is low, adiponectin is actually high. Those two balance each other, and you want that sort of ratio. IL-6 is low, and that's a condition for very low cancer development. This is an anti-cancer sort of profile that we've seen time and time again across a number of cancer types, including, in this case, this MMTV Wnt1 mammary model that we've developed. In contrast, the obese, they have a high IGF-1. Insulin is quite high and really out of control. Insulin resistance is evident. Leptin really rose, adiponectin's dropping, so that ratio has gotten quite high. And we see IL-6 levels quite high. And that's a recipe for rapid tumor development and growth. And we see that, again, across a number of tumor types in model systems. And the overweights are intermediate in both terms of tumor development as well as these markers. So we've got a panel of potential intermediates to go after. And the challenge is, how do we sort that out? And I'm a big believer in the power of epidemiology, but boy, I think this would be a big challenge to sort this type of, where they're such tightly associated. This would be difficult to sort in an epidemiologic approach. So luckily we've got the power of mouse genetics, and as well as the power of some pharmacologic agents to really pick this apart. And so a series of these genetic and pharmacologic approaches addressing which of these are really the drivers, which are bystanders. What we found was indeed this insulin IGF-1 part is a key component of this, and the inflammatory is another key component. I would call the adipokines more amplifying components, but what seems to be driving the growth and survival aspects of the insulin IGF-1 aspects. Now, these are receptor tyrosine kinases. That's how they function. The receptors for insulin, particularly insulin receptor A and insulin-like growth factor 1, signal through this pathway, PI3 kinase, AKT, mTOR. And not only is this a proliferative pathway, but it's also tied in very closely to protein translation. And it makes sense. You want that pathway linked to energy. You don't want to turn on a lot of new protein production in the absence of sufficient substrate. So not surprisingly, in the diet obese mice, and this is both a normal and tumor tissue across a number of tissue types, the obesity activates this growth and survival pathway. Not surprising, there's a lot of energy associated with the obese state, so that all systems go in terms of growth and survival kick on. Interestingly, when we look at the calorie-restricted mice, those are those lean animals, this pathway is really tuned down. We see how we really probe this is looking for phosphorylation of these kinases along this step. It's almost like a dominoes process into the nucleus that drives these processes, and calorie restriction or staying lean really drives this down. We were curious, well, what happens if we think from our genetic and pharmacologic approaches that perhaps IGF-1 and insulin are big players in this. What happens if we mimic the alterations in their receptor, either genetically or pharmacologically? And indeed, every time we manipulate downward the IGF-1 receptor activation, or insulin receptor A, or mTOR, which is downstream of these components, we see it mimic the calorie restriction in terms of this pathway as well as tumor development. And conversely, so when that's down, it mimics the low-calorie state. Conversely, when we activate that pathway, IGF-1 receptor, insulin receptor, mTOR, we mimic the obese exactly down this pathway to mimic their tumor response. So we think we've got maybe a candidate target here, this mTOR signaling pathway. And I think that we've had some proof of that principle. I think the clearest one comes from this experiment. Rebecca DeAngel was a grad student in the lab. So this is tumor weight, the same model we've been talking about, Wnt1, mammary tumors. Tumor weight in lean, control, and obese mice. And the blue bars reflect with, kind of also what we've been talking about, the absence of any drug intervention. The lean mice, the tumors don't grow very well. The obese, the tumors grow like crazy. And in the control, these overweight mice, they're intermediate. What happens if you bring in a specific mTOR inhibitor, and this is Afinitor or RAD-01, into the picture? And so when we treat even these lean mice where the tumors aren't growing very well, this is almost undetectable. It essentially shuts down tumor development when combined a low-calorie plus the Afinitor. In the intermediate group, addition of the mTOR inhibitor, and these mice are still overweight, their leptin's still high, insulin resistance is still there. But when we bring their mTOR signaling down, we can actually make them look like calorie-restricted mice, even though they're not calorie-restricted, by blocking mTOR. And I think the real interesting one was the obese. at one dose of the mTOR inhibitor, they're looking like the intermediate overweight mice in terms of tumor development. Again, these mice are still quite obese. They're about 50% body fat. Their leptin's sky high. Their insulin is out of control. But by blocking the downstream component, they don't look obese in terms of their cancer development. And a higher dose, and that reflects some of the resistance I mentioned in terms of chemotherapeutic agents. Took a higher dose, but when we block mTOR more effectively, we've brought the animals down to the calorie restriction level, even though they're still obese. This sounds too good to be true. This is the dream, right? I can eat all I want, take a pill, and no worries. And that's in a sense, what this is kind of saying. But I think what it tells us really is that perhaps obesity or the weight's not necessarily the target. It's the metabolic perturbations that are associated with the weight that are the key targets. And that's relief, because it's so hard to lose weight. And like I said, with 720 million people in the world obese, we're not going to get everybody back to their normal weight. That's unrealistic. But if we can disconnect the metabolic alterations that are linking the obesity and the cancer, well, then we've got a real shot at reducing obesity-related cancers. And so this is one encouraging target. And it looks good. What I'd like to say about mTOR, that's a common target in cancer therapy, and it's the success rates have been hard fought in that in terms of as a single agent. Certainly, in a therapeutic realm, it seems to work much better in the prevention realm. If we can keep this mTOR activation at bay, that is a strong anti-cancer, a preventive sort of approach, and so I'm enthusiastic about that one. So there's one target that we've identified. Now this gets to this transdisciplinary approach and trying to accelerate the pace at which we translate some of these findings from mice and in vitro studies as well to the human. So I work in this area: animal models, basic nutrition. And often I will find myself talking to clinical oncologists or epidemiologists, or behavioral scientists. But wouldn't it accelerate the process, we thought, if we're working on the same team, doing the same asking the same questions in our systems, using the same biomarker platforms and the same pathology and molecular biology and immunology and endocrinology sorts of approaches in the different modes of research, human, mouse. And that's exactly what we're trying to do. So we're trying to bridge this in a transdisciplinary fashion. And the key here is really modeling more closely the human situation, getting them aligned. There's a lot of, you can criticize the preclinical field, and people have, for the discordance between animal and human research, particularly in the drug development area where you're maybe trying to target a specific kinase. The success rate is not very good. It's in the teens, typically, in terms of what we find in a mouse translating to a hit in the human. Well, there's been a lot of discussion, how do we better align? And I think this is one approach, is to align them closely as a team and using the same markers. And I'll give you an example. Working with Dr. Carol Fabian at Kansas Cancer Center, who's a breast cancer prevention trialist and medical oncologist. Okay, thanks. And so Carol does these trials, and she's the queen of the fine needle aspirate sampling. So we've got access to target tissue in this sort of thing. And so we're aligning our mouse model studies with these trials. In this case, this was a weight loss trial using a low-calorie diet, pre and post fine needle aspirate, where you're actually getting breast fluid and breast cells from these women. These are not cancer patients. They're high risk, obese. And we're looking at serum in our lab, at insulin, cytokines, all the things we were mentioning. Carol typically looks at proliferation and mammographic density. And then we're also looking in the fine needle aspirate tissue markers, working with Gordon Mills at M.D. Anderson still on this, so with his reverse phase array approach, as well as other molecular and biochemical approaches in the FNA material. And what we found was again, with sort of modest weight loss, you don't see a whole lot changing. That's sad. Unlike diabetes, where you see a reversal with about 10% will really fix a lot of the ills of diabetes. It looks like it takes more weight loss to move many of these cancer-related markers. And so what we've got to do is get a little smarter about combining moderate weight loss with some targeted approaches to tune down that chronic inflammation and tune down that activation of mTOR activity. I did want to mention pancreas. This is not limited to breast cancer. We've seen a number of cancers, a similar story. In pancreas, because it's on the rise and because it is so deadly, as Margaret said, we haven't gone very far with our 5-year survival rates on this. We've got to learn how to prevent this one. So we went after, fortunately, most of the animal models available for pancreatic cancer are so aggressive, it's hard to do these sort of preventive studies. You know, it's hard to get a diet study in if the tumor's there in 6 weeks. That's too aggressive. So we worked hard on developing a relevant model that gives us time to get a diet intervention in and study it. So we have a Kras INK4A dual transgenic mouse here, genetically engineered mouse with sort of 2 components altered, the Kras oncogene and the INK4A tumor suppressor. And what's nice is you see the survival as a function of weeks on diet. So again, the calorie restriction really extended survival tremendously. The obesity accelerated in these overweight controls, intermediate. And what we found is, so we had time to get the diet in there and study it. And what we found is that obesity increases and calorie restriction decreases fatty pancreas and pancreatitis, which is a precursor lesion, a preneoplastic lesion for pancreatic cancer. Again, serum IGF-1 levels are higher, leptin adiponectin ratio, cytokines are higher in the obese. That signaling pathway is activated in the pancreas. And it's tied to NF-kappa B signaling, and that's something I didn't appreciate, was how closely linked the survival, mTOR survival pathway is to NF-kappa B mediated cytokine production. And so we have, I think, a dual target to go after there. So pancreas is similar. And in fact, there are, again, turning to some pharmacologic approaches, can we mimic the calorie restriction using drugs that target mTOR or target these inflammatory pathways? And a little proof of principle on this, we compared metformin, the diabetes drug, which is known to tune down the mTOR signal, and rapamycin, which is very potent, it's a direct mTOR inhibitor, and compared these 2 drugs in our pancreatic model. And both decreased tumor growth, both decreased mTOR signaling, particularly RAPA, the direct mTOR inhibitor. And it seemed to be through distinct microRNA-mediated mechanisms. So rapamycin really was all about mTOR and quite effective. Had no effect on insulin and glucose, had no effect on the stem cell population, really was driving down mTOR, and that seemed to be its full function. Metformin, in contrast, had a modest effect on mTOR, but it had broader effects on inflammation and the stem cell population. We saw decreased steroid formation in vitro. We see this miR34 mediated effect that drives EMT and some of the stem cell characteristics. So distinct effects of these, but again, I think a proof of principle that we can maybe mimic calorie restriction with some targeted agents. So I will stop there and with a sort of a take-home message. Yes, let's work on this obesity issue from the start. That's an important part. But hopefully by deconvoluting some of the mechanisms and mediators, we can go after particular targets, and mTOR is one, and this inflammatory process is another, and decrease cancer risk and progression. So I will stop there. Some of our future approaches, a lot of these are epigenetically regulated, as we've heard some about, and so that's a current focus of the lab. I'll stop there, hear my collaborators, and answer any questions you might have. Thank you very much. Okay. Okay. John, yes. Lots of my former bosses are here. It's good to see you again, John.
I wonder if it was a great talk. I wonder if you could address the issue of obesity being causative or stimulatory.
Yes.
And the animal data you showed suggest where they were all these were animals with cancer, and you reduced death rate. You didn't prevent cancer, you prevented death from cancer after you've got cancer. And if you talk about tobacco, we know why it causes cancer. Yes. It causes mutation. How do you think obesity causes cancer, and do you think your approach so far has shown it can be used to treat cancer, or does it actually prevent cancer?
Yes. So in my view, and I think the general view of the field is obesity really doesn't cause cancer in a classical way. It's not changing necessarily, it's not driving a mutation necessarily. But it does alter the progression. And so I would say that if we can slow it down to a point where you die of something else and not die of cancer, that's my definition, fits in prevention there. But in the classical sense, it's really a progression factor, a driver, not necessarily the root cause, like a carcinogen you might find in a tobacco exposure or something like that. Yeah.
Well, I agree with you so far, and I think articles like Dr. Huddis's article that say it talks about causing cancer, and it's dangerous. Because if we quit smoking, we're going to get rid of 30% of cancer.
Yes.
If we get back to being trimmer, we're still going to have cancer.
Yes, although I guess my sense and what we see in the animals is that we can delay the tumor development. So if you can delay it to the point where you're 110 years old when the cancer actually manifests and you're dying of something else in your 90s, then that's still a preventive, you're still preventing the cancer in my view.
Very good.
Yep, thanks.